Welcome back, my dear students. Today we are going to explore one of the most fascinating journeys inside our every living cell, that is glycolysis and Krebs cycle. The step-by-step energy pathways of cellular respiration, from the breakdown of a single glucose molecule in the cytoplasm to the generation of high energy ATP in the mitochondria. We will uncover how your cells extract and store the energy that keeps you alive and active. By the end of this class, you will clearly understand how glycolysis sets the stage and how the Krebs cycle completes the story of energy release, making life's process possible one molecule at a time. Glycolysis is basically a sequence of 10 enzyme catalyzed reactions that break down glucose, a 6 carbon molecule, into 2 molecules of pyruvate, which is a 3 carbon compound, and releasing energy in the form of ATP. Glycolysis occurs in the cytosol of the cell and represents the first step of glucose metabolism. This pathway is also known as Embden-Meyerhof-Parnas or EMP pathway and is divided into two phases, the preparatory phase and the payoff phase. So in this process of glycolysis, glucose 6 carbon compound is lysed into 3 carbon 2 molecules of pyruvate, and then energy in the form of ATP and NADH is released. Pyruvate further metabolized in energy pathways. Glycolysis has two phases: preparatory phase, that is the first phase, and the second phase is the payoff phase. So in the first phase, glucose is converted into glyceraldehyde 3 phosphate with the help of energy, and in the second phase, glyceraldehyde 3 phosphate releases energy and form two molecules of pyruvate. So what happens in the phase 1 or preparatory phase? In this phase, the cell invests 2 ATP molecules to prepare glucose for breakdown. It includes 5 enzymatic steps. Step 1 is the phosphorylation of glucose. Glucose is phosphorylated to form glucose 6-phosphate using 1 ATP molecule. The enzyme hexokinase, found in most tissues, or glucokinase, found in liver, catalyzes this reaction. This step is irreversible and traps glucose inside the cell. Then glucose 6-phosphate serves as a key intermediate in glycolysis and glycogen synthesis. Step 2, isomerization of glucose 6-phosphate. The enzyme phosphoglucoisomerase converts glucose 6-phosphate into fructose 6-phosphate. This rearrangement changes the structure from a 6-membered ring or glucose to a 5-membered ring or fructose. This reaction is reversible and it requires Mg2 plus ions. This is the first phase, glucose to glucose 6-phosphate with the help of hexokinase, and 1 ATP is consumed in this reaction. Then in the second reaction, glucose 6-phosphate is converted into fructose 6-phosphate. Then in the third reaction, fructose 6-phosphate with the help of phosphofructokinase, it will consume another molecule of ATP and it will form fructose 1,6-bisphosphate, and with the help of aldolase enzyme, fructose 1,6-bisphosphate will form glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Step 3, that is phosphorylation of fructose 6-phosphate. We know phosphofructokinase catalyzes the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate using another ATP molecule. This is an irreversible and rate limiting step in glycolysis. Phosphofructokinase acts as a key regulatory enzyme. It speeds up glycolysis when ATP levels are low and slows it when ATP is abundant. What happens? Step 4, that is the cleavage of fructose 1,6-bisphosphate. The enzyme aldolase then splits fructose 1,6-bisphosphate into 3 carbon molecules, glyceraldehyde 3-phosphate, that is G3P, and dihydroxyacetone phosphate, which is DHAP. This reversible reaction marks the beginning of energy extraction. So what happens in step 5? Isomerization of dihydroxyacetone phosphate. So the triose phosphate isomerase enzyme converts dihydroxyacetone phosphate into another molecule of glyceraldehyde 3-phosphate. This ensures that both resulting molecules can enter the next phase of glycolysis. This completes the preparatory phase, ending with 2 molecules of glyceraldehyde 3-phosphate. So now we'll go in the second phase, that is payoff phase. Now the glyceraldehyde 3-phosphate molecule is oxidized to generate generate ATP and NADH. This phase includes another 5 steps and produces 4 ATP and 2 NADH molecules, leading to a net gain of 2 ATP per glucose molecule. Step 6, oxidation of glyceraldehyde 3-phosphate. G3P is oxidized to form 1,3-bisphosphoglycerate or 1,3-BPG by the enzyme glyceraldehyde 3-phosphate dehydrogenase. During this process, NAD plus is reduced to NADH. This step conserves energy by forming a high energy acyl phosphate bond. What happens in step 5? That is the formation of ATP. First, substrate level phosphorylation occurs in this step. So the phosphoglycerate kinase transfers a phosphate group from 1,3-BPG to ADP, forming ATP and 3-phosphoglycerate. This is the first ATP generating step of glycolysis. Since two G3P molecules enter this phase, two ATP molecules are produced here. So this is the second phase or the payoff phase where energy is produced. Now we'll go at the step 8 of the second phase. Here the isomerization of 3-phosphoglycerate, the enzyme phosphoglycerate mutase rearrange the 3-phosphoglycerate to 2-phosphoglycerate. This reversible reaction requires Mg2 plus ions and dehydration of 2-phosphoglycerate. Then enolase removes a water molecule from 2-phosphoglycerate to form phosphoenol pyruvate, which is a high energy compound. This reaction is inhibited by fluoride ions. Step 4, formation of pyruvate. Second, substrate level phosphorylation. Pyruvate kinase then transfers a phosphate group from phosphoenol pyruvate to ADP, forming ATP and pyruvate. This is another irreversible step, producing 2 more ATP molecules per glucose molecule. So what is the net reaction? Glucose reacts with 2 ADP, 2 inorganic phosphate and 2 NAD plus. It will then produce 2 pyruvate, 2 ATP and 2 NADH. So that is the net yield of glycolysis is the 2 molecules of pyruvate, 2 ATP molecule, 2 NADH plus 2H plus and 2 H2O. So in summary, 2 ATP is used in the preparatory phase and 4 ATP produced in the payoff phase. So net gain 2 ATP and 2 NADH per glucose molecule. So what is the next step of glycolysis? After glycolysis, conversion of pyruvate to acetyl-CoA. We know at the end of glycolysis, 2 pyruvate molecules are produced. Each pyruvate then undergoes oxidative decarboxylation to form acetyl-CoA. Pyruvate is a 3-carbon compound and acetyl-CoA is a 2-carbon compound. So the 1 carbon dioxide molecule is removed out, which then enters and then acetyl-CoA, the 2-carbon compound, enters into the Krebs cycle. Steps of pyruvate oxidation, decarboxylation, that is 1 carbon is removed from pyruvate as CO2, leaving a 2-carbon fragment. Then the 2-carbon molecule is oxidized and the electrons are transferred to NAD plus, forming NADH. Formation of acetyl-CoA, the resulting acetyl group then attaches coenzyme A to form acetyl-CoA. This reaction is basically catalyzed by a large enzyme complex called the pyruvate dehydrogenase complex or PDC, which contains multiple enzyme subunits and cofactors derived from vitamin B5. Now we will start the Krebs cycle, which is also known as TCA cycle or tricarboxylic acid cycle. It's a biochemical pathway and it is used to generate energy through oxidation of acetyl-CoA. So synthesis of NADH, production of amino acids. So mitochondria of eukaryotes, and it happens in the mitochondria of eukaryotes and in the cytosol of prokaryotes. Citric acid cycle, it is a series of 8 enzyme catalyzed reactions that takes place in the mitochondrial matrix. Firstly, scientist Hans Krebs discovered the citric acid cycle, so this cycle is also known as Krebs cycle, and the first compound produced in this cycle is the tricarboxylic acid. So it is also known as tricarboxylic acid cycle. It is the central pathway of aerobic respiration, oxidizing acetyl-CoA to form carbon dioxide while generating NADH, FADH₂, and GTP or ATP. What are the steps of Krebs cycle? Formation of citrate. Acetyl-CoA combines with oxaloacetate to form citrate, catalyzed by citrate synthase. Then acetyl-CoA is released and the reaction is irreversible. Isomerization of citrate to isocitrate. The aconitase enzyme then rearranges the citrate to form isocitrate via the intermediate cis-aconitate. Then this shifts the hydroxyl group to prepare for oxidation. Oxidation of isocitrate to alpha ketoglutarate. Isocitrate dehydrogenase enzyme oxidizes isocitrate to alpha ketoglutarate and releasing carbon dioxide and producing NADH. This is the first decarboxylation reaction of the citric acid cycle or Krebs cycle. Now, conversion of alpha ketoglutarate to succinyl CoA catalyzed by alpha ketoglutarate dehydrogenase enzyme. Another carbon dioxide is released in this step and NADH is produced. The resulting compound is succinyl CoA. Then succinyl CoA is converted into succinate with the help of the enzyme succinyl CoA synthetase, and it releases GTP or ATP. This is an example of substrate level phosphorylation. Oxidation of succinate to fumarate. Succinate dehydrogenase converts succinate to fumarate producing FADH₂. This enzyme is also part of the electron transport chain. Hydration of fumarate to malate. Fumarase adds a water molecule to fumarate forming L-malate. Oxidation of malate to oxaloacetate. Malate dehydrogenase oxidizes malate to oxaloacetate generating NADH. The regenerated oxaloacetate then further re-enters the cycle. So this is the diagram showing the process of Krebs cycle, the reaction steps of Krebs cycles, and this is the results of Krebs cycles are the 3 molecules of NADH, 1 molecules of FADH₂, 1 GTP, 2 molecules of CO2, and it will ultimately produce 6 NADH, 2 FADH₂, 2 GTP and 4 CO2. Electron transfer chain will form the ATP. So energy yield from one term of the citric acid cycle, from one molecule of acetyl CoA, the cycle produces 3 NADH, 1 FADH₂, 1 GTP and 2 CO2. Since each glucose molecule produces 2 acetyl CoA, the total yield per glucose molecule is 6 NADH, 2 FADH₂, 2 ATP. These high energy molecules feed into the electron transport chain for further ATP production. In summary, glycolysis, we know it will from the glucose to pyruvate, 2 molecules of pyruvate, 2 molecules of ATP, 2 molecules of NADH is produced. After pyruvate oxidation, we gain 2 molecules of acetyl CoA, 2 molecules of NADH and 2 molecules of CO2. Then 2 molecules of acetyl CoA will enter into the Krebs cycle and it will produce 4 molecules of carbon dioxide, 6 molecules of NADH, 2 molecules of FADH₂ and 2 ATP. Together, these processes form the core of the cellular respiration, converting food energy into the usable chemical energy or ATP. So my dear students, we explored the fascinating journey of how our cells extract energy from glucose through glycolysis and the Krebs cycles, the two key stages of cellular respiration. We learned how glucose is broken down into pyruvate in the cytoplasm and how the acetyl CoA enters the mitochondria to fuel the Krebs cycle, generating vital energy molecules like ATP, NADH and FADH₂. These reactions together form the very foundation of life's energy flow. If you enjoyed learning about how cells turn food into energy, then don't forget to like, share and subscribe for more such engaging lessons in biology. See you in the next class, and as always, keep exploring, keep learning and stay curious. Thank you.